At the end of February, the Caltech Policy Center hosted a workshop on the pathways towards the safe and effective deployment of engineered microbial technologies. The goal was to foster the discussion on how to develop policy and regulation around engineered microorganisms, something that we do a fair amount of here at that we do a fair amount of research on here at Caltech. John Marken is a postdoctoral scholar in biology at Caltech, and you are one of the key organizers. John, for someone not so familiar with this sort of research, what are engineered microorganisms and why is it such an important area of research?
Yeah, so engineered microorganisms are essentially microorganisms like bacteria or fungi, anything that is a microbe, where you can genetically engineer these organisms in order to have them do things that are useful to human society. So people have been engineering microbes for a very long time, since the dawn of recombinant DNA in the 1970s. But the reason it's kind of become a very important area of research today in particular is because about a quarter century ago, in the year 2000, a field called synthetic biology emerged, led in part by the PhD work of Michael Elowitz, who's now a Caltech professor here, that essentially took the mindset of programmability from computer science and ported it into the genetic engineering space for microorganisms. So essentially brought in a new mindset saying, okay, we can think about the genetic background of a microbe as something that can be programmed. So we can create genetic programs and circuits of our own that can allow microbes to sense and respond to environmental cues in ways that we want them to do for particular applications, and essentially create almost biocomputers. And so you can imagine now we've transitioned from a more commodities production-based mindset where we were doing things like making recombinant insulin back in the 70s and 80s to moving into, okay, well, what can we do now with creating an autonomous biological agent that can go into an environment and do something such as sense the presence of a toxin in an environment and then on its own go and react to that by perhaps secreting an enzyme that can then degrade that toxin. And so about 25 years ago, the potential for this type of application was first unlocked. And over these past 2 decades, the technology has really developed and matured to the point where now I think we're starting to get into the space where people are really thinking critically about how do we take this technology that we've developed and apply that to pressing societal problems today.
So what do you think are the biggest areas of applications where we'll see genetically engineered microbes enter into society and use in industry?
Yeah, so one of the really exciting things about microbes is that they're very good with interfacing with the physical and particularly the chemical part of our physical universe. And so many of the big technological advances of recent decades, for example, the internet or today AI, are really more interfacing with the digital and the informational space. And so they've really transformed our society, but they've transformed it from an informational context. I think the microbial revolution is going to be a lot different in the sense that it can really change the physical structure and interface with the natural universe. So you can imagine, for example, things in agriculture or bioremediation. You can have microbes that are engineered to go in and really modify an existing environment, whether it's a controlled environment such as an agricultural cropland or a natural environment that's been contaminated by toxins. You can imagine microbes going in and modifying that for the better. You can also imagine more consumer-facing microbial products such as materials that are built out of microbes. One of the things that's really exciting is that we really have a very strong level of control over what we can have microbes do through genetic engineering. And so people are working on building fully functional structures out of microbes that can both have strong and resilient structural properties, but also include programmable sense and respond functionality. So you could, imagine something at the level of maybe there's microbes embedded in your clothes that can act as medical diagnostics by sensing your sweat and seeing if you have some, nascent disease coming up. Or you can imagine something like, vehicles that have a microbial coating that can prevent rust and corrosion, things like that. I think there's a lot of very exciting application areas, some of which we haven't even begun to think about yet, where microbes can really transform the way that our society interacts with the universe.
So we all remember the sort of debates and discussions around genetically modified crops. And now we are going to have genetically modified microorganisms. What are the sort of main differences for the regulation of crops versus microorganisms when they're genetically modified?
Yeah, so there are a number of biological properties of microbes that distinguish them from plants in a way that makes the regulation a lot more challenging. So for one, microbes are microscopic, so it's much more difficult to track and keep track of and find where genetically engineered microbes are in the environment. So that's just step one in terms of keeping them contained for testing and also for deployment. Microbes also reproduce asexually, so you can't do things like create microbes that have sterile seeds, for example, because they don't have seeds, they just reproduce. And so if you have the organism there, it's going to reproduce and it's going to spread and propagate, and you have to figure out how to capture that in a regulatory fashion. They also engage in a thing called horizontal gene transfer very promiscuously. So this is where microbes can transmit their genetic material horizontally to another microbe in the environment. And so horizontal gene transfer occurs broadly across many different kingdoms of life, but in microbes it happens all of the time. And so if you introduce a genetically engineered microbe to an environment, not only do you have to regulate the fact that the microbe itself that you introduced is genetically engineered, you also have to regulate the fact that, well, the genetically engineered DNA inside that microbe can then be transferred into a natural microbe in the native environment, and now you have a whole different combination of things that you didn't anticipate. What are you going to do about that? And finally, microbes do this thing called sporulation, where they can form spores that can then persist for thousands of years inside the environment in a very dormant state that can then resurface once conditions are more favorable. So, if you introduce a microbe into the environment, you're potentially committing yourself to a very, very long time scale of interfacing and perturbing that environment. And so what do you do about that?
So can you speak a little bit more about the risks that are associated both with developing and deploying genetically modified microorganisms?
Yeah, so the biggest concern I think that we have is we want to avoid a lot of the classic cases we see in, for example, invasive species research, where you have either intentional, for example, with the introduction of cane toads into Australia, or unintentional through things like how the kudzu plant has really overgrown large areas of the United States South, dominance of a natural ecosystem by a non-natural organism that then really hurts the natural ecosystem. And so I think when you add in two factors, one, the fact that we don't really have a good grasp and understanding of what invasive species ecology even looks like in the microbial realm, and two, the fact that not only are you thinking about introducing microbial elements that are perhaps not native to that natural environment, but also the fact that they are genetically engineered, which means you're introducing foreign DNA that is certainly not native to that environment. What are the unknown unknowns that we have to think about in order to prevent things like the cane toad situation or the kudzu situation from occurring in environments all around the world? And I think this is a combination of basic scientific research in ecology and microbial physiology that has to be done, in addition to really well-informed regulatory policies that balance these risks of these unknown unknowns with the potential benefits of being able to unlock the many possible benefits of microbial biotechnology in the environmental space.
So at the workshop that you organized just recently, we took a slightly different sort of approach to standard Caltech workshops and brought in, industry representatives, regulators, and policymakers, in addition to the scientists that are regularly at workshops here. And we had both a public session on possible regulation, as well as then breakout groups that focused on the ethics of genetically modified microorganisms, on the challenges for policy and regulation, and then also on the sort of scientific questions that need to be addressed in order to figure out what the right policy is. Could you describe a little bit what these discussions looked like?
Yeah, I think the discussions were extremely productive. Everybody came in with, I think, the similar core understanding that this is a really critical emerging issue with a lot of open questions and challenges that it's not really clear at first glance what's the right way to approach them. And so everybody came in with their different backgrounds and their different expertises, ready to contribute to this larger grand challenge, essentially. And I think it was very productive in the sense that we were able to speak across disciplines. We were able to kind of find common ground about what are the shared priorities that different people in different fields have, and how do we address these things. So I think it was very productive to have that, and I think it made for a different flavor of discussion than what you would normally expect at, for example, a typical academic conference. I think it really helped people of everybody who participated kind of break out of their typical mindsets and ways of thinking and see things in a new light that I think was very productive.
So in light of this workshop, what do you think are the sort of main challenges or possibly even opportunities for developing a robust policy on genetically modified microbes?
Yeah, so I think the fundamental major challenge with regulatory policy for this emerging space of engineered microbial biotechnology is the fact that many of these application areas are things that we still are just beginning to envision. So a lot of the current regulatory policy for biotechnology was developed with things in mind that we are very familiar with as a society. Things like growing food, that's something we did long before biotechnology, or producing commodity chemicals, that's something we did long before biotechnology. But now when you're starting to think about creating living functionalized materials, for example, that are going to be deployed out into the environment, or when you're thinking about creating autonomous microbial agents that are go in and monitor environments to kind of keep them at a particular healthy state, for example, that's something that is starting to breach into territory where there isn't an initial basis in a non-biotechnological way. So the fact that biotechnology is going to be the first step into these application areas makes it very difficult to write regulatory policy that can properly capture and the risks associated with doing so, while also promoting innovation and supporting the development of new science and new technology in this area. So that's a balance that's really going to have to be struck and will be challenging to do so.
Can you say a little bit about your own research and to the extent to which it connects to these sorts of policy questions?
Yeah, so I'm supported by the Resnick Sustainability Institute here, and I'm very interested in how engineered microbial biotechnology can be used to advance questions of sustainability. And I'm particularly interested in how engineered microbes can interface with terrestrial soil environments, for example, in promoting agriculture or also in promoting environmental remediation, for example. For me, in particular, I'm very interested in the pipeline of microbial product development from the laboratory into translation and deployment in an actual application context. And the questions of why is it that sometimes you can develop a product in the laboratory, it works really great in the laboratory, and then you take it into the field site and it doesn't work at all. And so there are many interesting questions about the interaction between microbial physiology and the behavior of a programmed genetic function, or the interaction between the environmental conditions and how the microbe acts, that I think are fundamental scientific questions that can help really enable new microbial technologies that are intended for translation to actually make it through that pipeline.
So if I remember correctly, it was sometime last summer, actually, that we sat down together for the first time to think about the possibility of this workshop. I had heard about your research, and it seemed sort of like the perfect fit for the sort of things that we want to do at the Policy Center. It's something where Caltech is actively doing research in this area, and it's obviously something, as you've described, where policy and regulation has to be developed, and is where that discussion is very active, and perhaps we could foster that sort of discussion here on campus. What made you interested in this sort of policy aspect of your research?
Yeah, so I guess it's funny you say that. It was actually, I went back and looked at my email. It was 1 year to the day that Neil Fromer emailed me. Neil Fromer, who runs the Resnick Sustainability Institute, and said basically, oh, I just had a chat with Mike and Frederick at the Caltech Policy Center. They're really interested in looking for spaces where the center can really make a contribution. We were brainstorming and thinking about GMO and how it can apply to sustainability. Can we find a time to meet and pick your brain about a more focused area where this could actually happen? And I think it's very exciting that, first of all, that I think, one of the things that's really exciting is like, at a place like Caltech, this is a place where I was just a graduate student at the time, and yet, I can be involved in the conversation at this very early stage. I think that's something really unique about Caltech, where we bring kind of insights from everybody in the community, even if they're just graduate students, and early on in the conversation to shape how these things are going to go. So that's a very exciting thing. And yes, I think the policy angle is really relevant, I think, to this emerging space because it's so emergent. I think a lot of scientists in this space are doing really exciting work, but they also have this understanding in the back of their heads that, well, right now, the things that we're doing are going to be useful for a potential downstream application that we recognize is currently infeasible under the regulatory landscape. And so the question is, well, what needs to actually happen to get all this amazing scientific work that's being built up and developed right now? How are we going to get that in a way that can actually benefit society? And really, it felt like advancing the conversation about regulatory policy was the major hurdle for this field in kind of letting it advance further. And so I became really interested in the policy space because I recognized that I can do all this science, but at the end of the day, if I want it to have an impact, I need to also make sure that the regulatory policy is, in a sense, catching up to the pace of this rapid development of the scientific technology. And so, yeah, I think that's really what drove me to the policy angle. Yes.
And what do you think are the sort of advantages of having that discussion here at Caltech?
Yeah, I think, for something where the regulatory policy is so intrinsically tied into the science, the conversation needs to happen at an institute that really understands the science very well. This is not the kind of conversation that can happen without scientists and innovators and developers in the room with the regulatory people and the policy people to make sure that the conversation is happening across disciplinary boundaries and making sure that perspectives that may not be well recognized or well valued in some disciplines but are in others are all being considered equally. Because I think this is a very big and very grand challenge, and we're not going to be able to resolve it in a useful and effective way without having everybody's input.
John, many thanks for taking the time to talk with us.
Yeah, thank you.